All right, well, I think we'll go ahead and get started. It's a pleasure to welcome Dr. Kim Rathmell, who's giving an invited Cancer Center seminar here at Brown University. I wish we were doing this in person, and as we were saying earlier, we hope we don't get Zoom bombed today, but Dr. Rathmell is the chair of the Department of Medicine at Vanderbilt, where she's also an endowed professor of medicine and biochemistry, a GU medical oncologist at Vanderbilt University School of Medicine. She earned her MD and PhD in biophysics at Stanford University, and at that time, she did some work with Gil Chu and published some nice papers in the area of DNA repair. Then went to the University of Chicago, medical internship, then continued with her residency training at University of Pennsylvania and did a fellowship in medical oncology at University of Pennsylvania. She did postdoctoral work with Celeste Simon at Penn and Terry Van Dyke at UNC, and she held faculty positions at UNC before moving to Vanderbilt as director of the Division of Hematology and Oncology in 2015. Currently, she is also the physician-in-chief at Vanderbilt University Medical Center and deputy director for research, integration, and career development at the Vanderbilt Ingram Cancer Center. She has had a distinguished career as a physician scientist with leadership roles such as president of the American Society for Clinical Investigation in 2019 to 2020. She was elected as a AAAS fellow. She's currently PI of a T32 in molecular oncology and a K-12 grant. She has served on ASCO ethics and education committees and has led working groups with
you have called is not active. For more information, please visit www.telecomiq.com.
Right. Okay, so in addition, she has gotten an AACR Team Science Award. She has various editorial board roles with journals such as the JCI, Cancer Research, JCO Precision Oncology, and has been serving on the Board of Scientific Advisors of the NCI. Dr. Rathmell has published over 200 publications on various topics related to kidney cancer, models, mouse models, screening, epigenetic regulation, metabolic features, and novel therapeutics. And I think we're going to hear quite a bit about that today. In 2021, she published a paper in Nature entitled Cell Programmed Nutrient Partitioning in the Tumor Microenvironment. And also this year, she's had a couple of papers in JCI Insight on one on neoadjuvant pazopanib in renal cell cancer. She had earlier in her career published on sorafenib in the neoadjuvant setting in renal cell cancer. And she had the second paper in JCI Insight this year was about hypomethylation leading to increases in transposable element expression and immune signaling in kidney cancer. She's written about a variety of other topics of interest to physicians and physician scientists, including conflict of interest, drug shortages, impact of the COVID pandemic, and physician scientist careers. So it's really great to have Dr. Rathmell with us today, where she'll be speaking about metabolic adaptations in renal cell carcinoma and other tumors. And I'll ask you to unmute Dr. Rathmell and go ahead with your presentation.
All right, thank you so much for the kind introduction. Yes, we're a little bit all over the place. We like to tackle a problem from whatever edge we can get. But today we'll talk about metabolism. But you left out my most formative time, which was my month at the VA with Dr. Aldiria as my attending. So I learned really all my oncology right there. So with that, let's get this to go forward. Okay. I have a few conflicts. They're almost all my spouse, who's gotten himself interested in starting companies. So none of them relevant to this work. So the goal is to look at a hypothesis that metabolic demands are programmed features of cells in tumors, and that this presents a vulnerability that we can exploit. In the future, we'd like to get to how we can do that therapeutically, but I think it'll be immediately obvious how that might be used diagnostically. Okay, so I'm going to start with a couple of cases, and I'm going to minimize all of you and me so that I can see all my screen. So, so the first, these could be patients of mine. I currently only see hereditary kidney cancers, but they're not. I chose them because they're published, and so you can look them up if you're so interested. The first is a case, and you'd think kidney cancer, we must be talking about 65-year-olds, but we're not. We're going to talk about young people. 22-year-old here who presents with hypertension, has a suprarenal mass, an elevated VMA, and has a PET scan. And in that PET scan, the adrenal mass is lighting up hot, and that's all. But he has a contrasted CT as well, and that shows that there are multiple hypervascular lesions in the kidney cortex, none of which are lighting up on PET scan. So just think about that case for a second. Okay. And in contrast, here's a second case, also a published case, but in this case, a similarly young person, a 24-year-old woman referred with abdominal discomfort, bloating, anorexia, and weight loss, has a very large renal mass, 45 centimeters. It's a complex cystic mass that's encompassing most of her abdomen, and you can see this mass here. It's intensely avid on PET scan. She's also got anemia. Okay, so we have two patients who have very young, presenting with tumors in the kidney, who have intense PET avidity in one case in the kidney tumors and PET cold in the other. And this has been something that's bothered me for a very long time. And so I'm really excited that we're finally having the tools and the ability to really tackle some of this. So before we tackle this, I think I use kidney cancer as my usual model system. And so I think it's important to know the model we're working from. Our tumors come from the kidney, so what do we know about the kidney? It's a really interesting place for cancer to start. So it's a unique environment of gradients. So there's glucose gradients at the cortex. It's very glucose replete, and at the inner medulla, it's very deeply.
Hello.
Hello, Kim. She looks frozen.
Frozen. That's
Oh,
tough.
boy. Let's see if we can call her.
I hate Zooms.
All right, this is not good. Okay, so she must hopefully.
I don't know what happened. Sorry about that.
You froze. You froze.
Yeah.
I was just going to try and call your office, but we're coming back.
All right, so you can see and hear me now, and
Yes.
I will try that again. I think we were in the gradient of the kidney. Does that sound right?
Yep.
Okay. All right. So this is a really great place to study tumor metabolism because the basal setup for these cells is different than in other places. So I want to give you a little bit of history of how how we got to where we are in understanding the hereditary kidney cancers. This is a picture of Burton Zabar, who was at the NIH for many years and was very interested in these kids and young adults who developed renal cell carcinomas and other kidney tumors, and set about in really the classic style, going around the country, finding families where there were collections of kidney tumors, and then testing the blood and doing old school genetics to identify these tumor syndromes and then the underlying tumor genetics. His partner in crime there was Marston Linehan, who's still the head of the urologic oncology branch at the NIH, a urologist. And together they really early on revealed the stratification of hereditary genetics, but it applies to somatic tumors as well. We know now clear cell renal cell carcinoma, that's the classic. When we say renal cell carcinoma, that's what people really mean, was linked to the VHL gene, loss of the von Hippel-Lindau gene, and we'll talk a lot about that biology. But others, mutations in MET, mutations in fumarate hydratase, so we'll dip down into the Krebs cycle, and mutations in the Birt-Hogg-Dubé gene. So these give rise to very different tumors, but all coming from the cortex of the kidney. Okay, so thinking back to our cases. So we had patient number 1 had a pheochromocytoma and a bunch of renal tumors, all PET negative, and patient number 2 had a massive renal tumor, very intensely PET avid. So just think for a minute, they're both young. Do we think this is sporadic renal cell carcinoma? Is it VHL syndrome associated renal cell carcinoma, FH syndrome, BHD, or CMET? And I won't had it set up at one point to see what people think, but I'll tell you that patient number 1 had VHL syndrome and patient number 2 had FH syndrome. So let's think about those two discrete tumor types. And they're really interesting because they both come from the proximal tubule. We are not able to identify a distinct cell of origin for these two tumors, but they're really, really different. So we know the most about VHL mutant renal cell carcinoma. We know that it's driven by high levels of HIF. We know that it's a very angiogenesis dominant disease. There is a metabolism component as well. We'll talk to in just a minute. But in papillary type 2, seemingly same cells at risk, mutations in the FH fumarate hydratase gene, as a byproduct, also upregulates lots of HIF. And it's a very metabolically dominant disease. Really, really different. Clear cell renal cell carcinomas in VHL will sit and you can watch them for years and years and years. We actually follow what we call the 3 centimeter rule. We don't do anything with them till they're 3 centimeters in size. Papillary type 2 renal cell carcinoma with FH mutation. I follow these patients. Well, we do scans every year, and that's not often enough. Once the tumor emerges, it becomes metastatic almost immediately. So really completely different biologies here, in addition to metabolic distinction. So a little bit more about the diseases. The VHL disease associated with hereditary carriage of the VHL gene mutation. In addition to renal cell carcinoma, they get renal cysts, they get hemangioblastomas, a very vascular tumor in the brain, as well as in the spine and the retina, and they get pheochromocytomas. HLRCC, it's called hereditary leiomyomatosis in renal cell carcinoma. It's biallelic mutation in the fumarate hydratase gene. You carry one and have loss of heterozygosity. So they get these massive, very aggressive renal cell carcinomas, and the leiomyomatosis. They get classically it's uterine leiomyoma tumors, and they have a leiomyoma of the skin as well. So they have a classic skin finding called a fibrofolliculoma. So the common link is dysregulation of the oxygen sensing syndrome. There's obviously some big differences. This was the subject of the Nobel Prize, and they call out Bill Kaelin. 3 people won the Nobel Prize that year, but Bill was really the one most focused on renal cell carcinoma. And the Nobel Prize was won for discovering this rheostat that measures low oxygen, and that's important for response to hypoxia. But in the case of kidney cancer, it's constitutively a hypoxia type defect. So I'll go here because the picture's a little bit better. So building the model of VHL-driven renal cell carcinoma, normally VHL, if it's normal and intact and functioning, it binds to a protein called HIF. HIF is made at a pretty rapid clip, but it binds to VHL after an oxygen-dependent mark is placed. So in the presence of oxygen, binds to VHL and is degraded by the proteasome. So you don't get very much HIF, even though it's made really fast. And so if you lose oxygen, you can very quickly bypass your VHL mediated degradation and make very high levels of this protein. Or you can just lose VHL, like the case of clear cell renal cell carcinoma, and you have high levels of HIF constitutively on. So it's a transcription factor. It partners with a heterodimer partner, HIF-1 beta, and it turns on a host of genes. So more than 1,000 genes have been ascribed to hypoxia-inducible factor programming. This picture just shows some of the metabolic features. So it turns on a whole bunch of transporters and enzymes that are important for driving metabolism. So here's GLUT1, and should push glucose into the cell or allow glucose to enter the cell. LDHA to drive pyruvate to lactic acid. PDK1 actually prevents pyruvate from entering the mitochondria and creating substrate for the TCA cycle. But it also increases genes involved in glutamine uptake. So there's a lot of metabolism work that's been happening in trying to understand how cells respond to low oxygen. And this makes sense. If cancer aside, if you don't have enough oxygen, you want to change the way you do metabolism. You no longer want to be so dependent on an oxidative phosphorylation. You want to switch to glycolysis. And in this case, it's aerobic glycolysis because there is plenty of oxygen around. In the case of FH, it's a little bit different. Oh, oops, too fast. In the case of FH, it's a little bit different. FH is one of the key enzymes of the TCA cycle, and so you lose FH, and that uncouples that cycle, and it does two things. One is it leads to an accumulation of alpha-ketoglutarate, and that alpha-ketoglutarate can poison the prolyl hydroxylase, and so you get upregulation of HIF. As I said, it's as a byproduct, but it's a lot of HIF can be made that way. And by uncoupling this TCA cycle, all the glutamine that's coming in or the glucose that's moving into this cycle can't drive a functional TCA cycle. And so instead, the cells use those substrates to undergo reductive carboxylation, which isn't inefficient, but a way of making energy. So this whole concept of using glycolysis in the absence of, or with plenty of oxygen around, is what we call Warburg metabolism. And Otto Warburg won the Nobel Prize for this. And he recognized that cancer metabolism was different than normal metabolism, and that tumors could take up more glucose than a normal cell would, even in the presence of oxygen. And it's on the basis of this that we use PET scans. So we use lots of PET scans in cancer. So it's F18 labeled 2-deoxyglucose. That 2-deoxy means that it after the first step in glycolysis, it can't continue to proceed, so it gets trapped in the cells. And so it's only cells that are taking up lots of glucose that will light up with F18. And so you can scan a patient who has cancer, in this case, I think it's a lung cancer, and see where pockets of cancer cells are. Okay, so that's all great. So what's the Warburg phenomenon? If we break it down a little bit more, it's by comparison to normal, where glucose enters and undergoes glycolysis, you make pyruvate, enters the mitochondria, uses oxygen, makes CO2. That's how you make energy. There's a little bit of lactate that gets made alongside. But in the setting of Warburg metabolism, glucose is taken up in abundance. You make lots of pyruvate. It does not go into the mitochondria, it makes lactate instead. So this is Warburg effect, classically. So I just told you that VHL is lost in renal cell carcinoma. You make lots of HIF. It turns on all these enzymes. This should be classic Warburg, but I started by showing you a PET scan that was negative, and VHL patients classically have negative PET scans on their kidney tumors. So what does that mean? So this is something that has literally bothered me since I was a fellow. There are a lot of different reasons. Let's start in the cancer cell. So first off, what we early on, back when I was a fellow, understood best was that VHL turned on HIF, and we really knew most about HIF1. But back about that time, we recognized that there were more than one HIF factor. So VHL loss can lead to accumulation of both HIF1 and HIF2. So very important to know which HIF is upregulated in these cancer cells, because HIF1 has a preference for glucose metabolism genes. HIF2, while very similar to HIF1, often doesn't hit those same glucose metabolism genes. It tends to hit more genes that are involved in dedifferentiation. Both hit angiogenesis. And so that was one reason for some of the confusion about these tumors. But we know that some tumors will express HIF1, some will hit HIF2. We actually looked at this as I was finishing up in Celeste Labs. This is an old paper. We took tumors and we were very curious about this correlation between HIF1 and HIF2. There's a small subset of clear cell renal cell carcinomas that have VHL wild type, and they have no overexpression of HIF1 or HIF2. But of those that are VHL mutants, they split down. It's not right down the middle. It's actually more of them will express just HIF2 than HIF1, and some will express HIF1 and HIF2. And so, so we thought at the time that these would be more you're metabolically active, and these would be metabolically different. Both would be angiogenic. And the plot gets a little bit thicker. This is work from Bill Kalin's group that showed that even sometimes when you have HIF1, the cells really don't like it. Turns out having too much HIF1 is really pretty toxic to cells. And so in another paper, he showed that overexpression of HIF1 acted negatively in cell proliferation. But here, analyzed a bunch of different tumor lines that we had characterized as HIF1 and HIF2 positive. We had the HIF2s and we had HIF1 and HIF2s. And when he did some allelic analysis, it turns out, although they stained positively, like over here, so protein, you could see some HIF1, it turns out many of them contained microdeletions. And so they were in-frame microdeletions in HIF1, so they were not functional HIF1. So the vast majority of VHL-driven tumors only use their HIF2. So we thought the story was done there, and did a little bit more. Just I will say that we can recreate that in some model systems. Here we used mouse embryo kidney cells that we engineered to stably overexpress only HIF1 or only HIF2. So now we don't have to worry about VHL anymore. We just have one or the other. We don't have both. And we saw that if we overexpressed HIF1, we got this is seahorse assay. So we got loads of glycolysis, oops, down here. And we got bunches of oxidative phosphorylation as well. So HIF1 overexpression did exactly what we had expected it would do, and HIF2 did not. HIF2 instead here, HIF2 overexpression maybe even decreased glycolysis, but mostly didn't have a very big change. But they still did a bunch of oxidative phosphorylation. So how do you skip glycolysis and move to oxidative phosphorylation? I think I have one slide. Hopefully I didn't take it out. Oh, I think I took it out. It turns out these HIF1s really like glucose. They'll eat glucose or glutamine. These HIF2s really, really love glutamine. So So it could all just be the cancer cells. Let's go back to doing some PET scans on people. We took some sporadic cases of clear cell type renal cell carcinoma, and we did some MR PET imaging. We used MR because you get better spatial resolution, but the PET is the same. It's an FDG PET scan. And I'm sorry, it cut off here at the top. This is a gradient of low PET avidity to high PET avidity. We could map within the tumor regions that were highly avid and regions that were low avid, and then take those samples and take a look at them. We did some transcript, sort of early radiomics, and we did some protein analysis. And it turns out those that were most PET avid had lost FBP1, a protein that is in the early stages of glycolysis, and that Celeste Simon had shown already is often lost in clear cell renal cell carcinoma. And that also correlated to high levels of GLUT1. But HIF1 really didn't correlate. And actually, there wasn't a lot of HIF1 here at all. So this kind of fit our model, that it was the metabolism of the cancer cells was largely favoring a less metabolic, less glycolytic feature set. Okay, so summary there, tumors displayed altered metabolic properties at the global functional level and tissue level, but in the cells themselves, they're shifting their metabolism depending on their genetic features. And here I mean HIF1 versus HIF2. And that classic Warburg effect may really require high levels of HIF1 and maybe even other metabolic uncouplings like the FH-driven tumor can do. Okay, but let's come back to what PET scans detect. So we think about PET scans as picking up cancer, but we all know that they pick up a lot more than cancer. I actually just got my booster yesterday, so my arm is exactly like this patient's arm. The PET scans pick up inflammation too. Rheumatologists know this, we oncologists know this, we see sarcoid and other things lighting up. And here's a patient who got a SARS-CoV-2 vaccine in their deltoid, and the next day happened to get a PET scan, and lo and behold, there's a bunch of glucose uptake here too. So immune cells use glucose. So how does this all fit in our cancer model? And so, but this is about the time I moved to Tennessee that we realized we better start looking at the immune cells in those tumors. So this is the people who have been involved in this. Most notably is Jeff Rathmell, my husband of more than 30 years, who I didn't collaborate with until 5-6 years ago. He's a tumor immunologist, so handy that. So, and then cross mentored in his group is Katie Beckerman. She now has a lab of her own at Vanderbilt. Peter Siska has a lab of his own in Regensstrafe in Germany. And these are two MD PhD students. This is not, that's Jim Allison, but Brad Reinfeld, who's in my lab and Jim Allison's band, and Matt Madden, both MSTP students. So this group started saying, what's going on in immune cells? Okay, I'm going to do a little bit of immunology here and talk about T cells. So this is all work that's actually from Jeff Rathmell's lab, and this is what got us going. So T cells have to convert from resting, they get stimulated. That's a period of anabolic, that shifts from oxidative to anabolic metabolism before they'll move on to become a memory T cell. This is what Jeff's lab has been studying for the last 20 years. And the nuts and bolts of it are that T cells are very picky about which fuel source they'll use, and the fuel source can drive T cell differentiation. So here's just a couple of examples. I should put them all both at the top. So if we overexpress, if Jeff's lab overexpresses GLUT1 in T cells only, what he'll find is that kidney sections will have deposition of antibody, so creating a autoimmune kidney disease simply by overexpressing GLUT1 in those cells. So the T cells become pro-inflammatory just by overexpressing GLUT1. If you knock out GLUT1 in an IBD model, so normally cells, because or mice who have this IBD model will lose weight because their IBD starts to flare. But if there's no GLUT1, those T cells won't engage, and so you don't get weight loss and death from IBD. So this trigger of glucose metabolism in T cells is really important. You can manipulate that not just by knocking out or overexpressing GLUT1, but by changing the drivers of other metabolites like glutamine. So glutamine is an essential amino acid. And so if you block the cells from being able to take up glutamine, they will switch and take up more glucose. And so here is a CB839 is an inhibitor of glutamine synthase. And these are FACS plots showing more activated T cells just because you gave them CB839. And here's, it's similar to if you knock out GLS, so you can either use the drug or you can knock out glutamine synthase. And here TH1s shown in red. This is activation, will activate more, as you see in red, if you knock out glutamine synthase, not so much with TH17s or Tregs. So, so you really shift the T cell repertoire. And so this was the conversations happening in my dining room table, right? Like me saying like, oh, it's all the cancer cells and they're reprogramming their metabolism, and him saying like, oh no, it's all about the T cells. And it turns out they're living there in the same space, just like us. And so we got interested in what's this communication happening between the cancer cells and the T cells, both of which are dependent on GLUT1 and glycolysis and potentially Warburg type metabolism. So we know that there's a lot of heterogeneity in tumors. There's hypoxia, like we get from our overexpression of HIF. There's pseudo hypoxia, but there's real hypoxia just because of outstripping blood supply. But there's potentially, the model was that there's competition for glucose or amino acids, or maybe there's too much waste product. And so the idea has been that the cancer cells are creating this toxic environment, and that's making the T cells really unhappy. And so we decided we would try to figure that out. So it turned out kidney cancer was a good place to do that. So kidney cancer, renal cell carcinoma, has the most CD8 exposure of all of solid tumors. So here's just the TCGA gated, looking for CD8A expression in these tumors by transcript analysis. So there's lots of evidence of CD8 expression, unlike other tumors where there's there can be these pseudo lymph node type organs. In renal cell carcinoma, you get just dispersed T cells. So it was kind of the perfect tumor type to study T cell metabolism in. Very convenient and fun for us. So I'll also remind you that this is suddenly important. So in 2015, we had the first immunotherapies beyond hydroxyl-2, so the checkpoint inhibitors approved for renal cell carcinoma. It's been transformative, and we want it to be even better. So I'll remind you that they can work really well with this patient. This is a patient of mine, had a big renal mass, multiple liver mets. This guy was 42 years old. We put him on single agent nivolumab. This is early days, 2015. And after a year said, you know, your tumor's not very big and I haven't been able to see your liver tumors in a really long time. And if you looked like this the day I met you, we would have just taken out that tumor. And we sent him to surgery, and this was now 3 years ago. He's been off therapy. And so this is how you cure patients with immunotherapy. And that's fantastic. But not everybody responds this way, even when their genetics seems really very similar. And so we really wanted to understand how we convert more patients to be responders to immunotherapy. And so we felt like we had to understand what's going on down there in the tumor microenvironment. So just to remind you, we've got these kidney cancer cells, VHL loss, doing whatever it is they're doing metabolically, and T cells that are, if they're preferentially using glucose, they're TH1s. If they're glutamine avid, they're more TH17 memory. And so we set up what we call the RCC Analysis Lab. We've now analyzed more than 450 tumors. Basically, patients present to the OR. We work with pathology and access their tumor, a blood sample. We separate that tumor. We now make organoids as well, but separate that tumor into a single cell suspension, perform metabolic assays. We get a pretty nice set of demographics and we store it as well. So this has become our gold mine, is to finally really work in patients. So first thing that we find is we decide to look at these T cells. And this is a CyTOF experiment, just trying to phenotype these T cells. Here we're showing from peripheral blood control resting T cells. Here's CD4s, here's CD8s. You can see they mostly gate in these smaller areas. And if you stimulate them, they reliably shift and their phenotype is more of a stimulated activated T cell. Both CD4 and CD8 do this. If you take TIL, tumor infiltrating lymphocytes from those same patients, what you'll see is that their CD8s and CD4s are phenotypically distinct, and they don't activate the same way that the peripheral blood in the same patient will. And this just shows what their phenotype is. And when we look at their activation, do they activate at all? They do. I show this sloth from a movie just to really, they'll go, but they just barely go. So here's an example. CD8 controls, if you activate these with CD3 stimulation, CD25 is a marker of stimulation. So they hop right over. Same for CD71, they increase cell size. Right? This is what you're supposed to do. This is what you get if you take a T cell straight out of a tumor. So a little bit. Same thing over here. We're looking at proliferation. Here's the controls. They just proliferate away. If you take peripheral blood from a patient, proliferate away. If you take TILs, not so much. So we had suspected that these cells were unhappy. And we showed that they really didn't stimulate the way cells normally will with just CD3. Katie Beckerman published a paper showing that we could push them to stimulate if you give them co-stimulation. So CD28 co-stimulation will activate and shift their metabolic features as well. This is a very busy slide, so I won't go through all of the data, but suffice it to say that co-stimulation will push them metabolically into the zone of a normal cell. So this is important for how we treat patients. We don't use anti-CD28 now, but there's a growing range of checkpoint inhibitors that are being used in combination that can really stimulate, can overcome whatever metabolic problem these cells have. So we wanted to know, what was that problem? So why are they failing to activate? At first, we thought it must be this competition. The cancer cells are hoarding all the glucose. And so Brad Reinfeld took a look at what we called the tumor juice, the interstitial fluid, and looked at it by mass spec. And this is normal versus in a normal kidney versus renal cell carcinoma. And there was really no difference in the amount of glucose. There is a little more lactate. So maybe it's a waste product, or lactate can be used as a fuel source. We're still investigating this, but it's not because they've run out of glucose. Have they run out of essential tools? So in gray here is control, in white is the TILs. No, they have plenty of GLUT1. They have plenty of hexokinase 2, the first rate laminating enzyme. They have plenty of phospho-S6, in fact, more than the controls. So why are they not moving? It turns out that the defect that these have has manifested themselves, this is probably multi-levels down, in disordered mitochondria. So their mitochondria have undergone fission, and this control, green is mitochondria. Normal mitochondria are fused and nicely organized together. Here's what you get with CD8 TIL, very fragmented mitochondria. So these mitochondria are not able to do oxidative phosphorylation the way you expect. We can see that on electron microscopy as well. control mitochondria are nice and large. These CD8 TIL mitochondria are small, little rounded mitochondria, and they don't function right. So they're hyperpolarized. This is a polarization assay. So the CD8 TIL are skewed far to the right compared to controls. And I'll just show you here, they also express higher levels of mitochondrial ROS and decreased levels of cytoplasmic ROS. So can we overcome this metabolically? So, so we know that you can co-stimulate, so you can push, and make them reprogram, but can you use metabolism strategies to overcome this metabolic blockade? And you can. So we had shown that there was lots of mitochondrial ROS in these cells. This is a schematic T cell, which we thought was receiving some negative signal from the cancer environment. And if we give a mitochondrial scavenger, like MitoQ, you can see over here in green is the MitoQ, these cells that really didn't want to stimulate now will. So CD25, CD71. So you can scavenge out that ROS and make those cells happier. You can also just bypass this, these early steps of glycolysis and just feed those cells pyruvate, and that will also push these cells to activate. And then finally, we had shown, I had shown you before data from Jeff's lab that if you blocked glutaminase with a drug, that those cells would shift to take up glucose instead, and that they would change their phenotype. And so you can do that here as well. You can take these TILs, hit them with CB839. They will increase their glucose uptake, which I didn't show you. Sorry, that should be here. And they also increase their activation, shown here with granzyme B. So, and this, sorry, that is published. That's Katie Beckerman's work. So to summarize, in this microenvironment, we know glucose is available, but the tumor infiltrating lymphocytes, especially CD8s, which we focused on, will fail to activate without further co-stimulation. They have plenty of mitochondria, but they're small and fragmented, and that the T cell activation can be partially recovered by metabolic interventions. So we think that metabolic defects are contributing to the poor T cell function in these tumors. Okay, now, but what is happening in the tumor and who is getting the glucose? So we went about this originally thinking we were asking the question, are the cancer cells or the T cells really taking up the glucose? We thought we knew it from the PET scan work. We thought we knew it from the T cell work, but it turns out we didn't know anything. So, so if T cells require glucose, is it available in tumors was the question, and who's winning in the competition? What we hadn't expected is that in addition to cancer cells and T cells, there are also myeloid cells. Okay, so I'll remind you that although there is some data that glucose can be limiting in tumors, that is not always the case. Here's another example of some studies in clear cell renal cell carcinoma patients where we're measuring glucose lactate. Here's glutamine, actually very high, but glucose here in tumor versus normal is not limiting. So at least in clear cell renal cell carcinoma, there's plenty of glucose around. So to do this, we did an old school experiment. Here's Brad and Matt, hard at work in the radioactive chamber. We took F18 2-deoxyglucose and did a traditional PET scan on mice that had injected tumors. So this is a PET scan just like you do on a patient, but now we have PET tracer that we can follow, and we can say which cells are actually taking up the PET. And so the tumor cells get fragmented from these tumors. And we see, first off, that there's a lot of different kinds of cells in here. So compared to spleen, we used a variety of models because we have to have an immune intact animal system. So MC38 is a colon cancer line, and you can see that there are some T cells, but there's everything in red and pink, and orange and yellow is more myeloid. So there's more myeloid than T cells, and B cells in green. Renca is a renal carcinoma, spontaneously arising kidney cancer line, and also lots of myeloid cells. This is a GEM model induced with polyoma middle T, also has lots of myeloid cells. So cancer cells have lots of myeloid, which is, I think, often forgotten. I'm just going to put these all up here. When we looked at where did the glucose go, we're showing here per cell basis or per million cell. Although you can detect it in the tumor, there's not proportionally more in the cancer cells. It's mostly showing up in the immune cells, okay? 2 different colorectal models, a kidney model, a breast model, and a bunch of other models. It was always showing up in the immune cells. When we break the immune cell compartment down even further, and this is really just a control showing the purity of these models. If you look at the bar graph on the right, the glucose is going, again, some to the whole tumor, but not so much to the cancer cells. That's the CD45 negative, but it's showing up in CD45 positive. And then further, if you enrich for F480, which is a macrophage component, it's here, not in the T cells. So we have macrophage uptake of glucose really outcompeting the T cells on a per cell basis. Are they just hoarding up that glucose because they're macrophages? No, they're using it. So we did seahorse assays on F480 macrophages compared to CD4CD8 T cells compared to tumor cells. So here in red, you can see they're doing more glycolysis and oxygen consumption. That's measured out here. And if we do gene expression for on these purified fractions of cells, these cells separate based on their metabolic profiles. So you can separate the myeloid cells from the CD4 and immune cells from the tumor cells. The CD45 negative cancer cells have most enrichment metabolically for amino acids and nucleotide and fatty acid synthesis or fatty acid metabolism, whereas the macrophages really are more glucose, carbon sugar metabolisms. So we took a look at some of these downstream targets. So this is transcript targets that were guiding us toward, is this a glucose consumer or an amino acid consumer? Hexokinase 2 is an important enzyme in glycolysis. You can see that it's highest in the TAMs, but its expression is driven by mTOR, so not surprising, and you hit with rapamycin and you can bring it down. CD98 is an amino acid transporter, and it's upregulated in the tumor cells, also mTOR dependent. So there's an mTOR dependent fixation of whether these cells are glycolytic preferred or amino acid preferred. So when we look at glutamine uptake, then by doing, we can do an FDG PET scan, we can do an F18 glutamine scan. And we did the same kind of F18 glutamine uptake. And just like the transcript data suggested would be the case, the glutamine, instead of going to the CD45 immune cells, went to the CD45 negative cancer cells, regardless of model. And here we're just showing, here's what these PET scans look like. That's an autorad of these cells. And I'll just skip you down here to this bar graph. There's the glutamine is showing up in CD45 negative, which we've shown is the cancer cells, not in the macrophages so much, or the T cells. Lipids is also something that we're interested in, and I will just show you that the CD45 negative, the cancer cells, again, also really like lipid much more than the macrophages or the CD3 positives. And just like what we had shown before with the T cells, if you block glutamine uptake, you can shift cells to preferentially take up glucose. So V9302 is an inhibitor of amino acid transport. And so if this inhibitor blocks glutamine uptake on the left, this is just a control. So you can see glutamine uptake using F18 glutamine inhibitor shuts that down. Okay, so that's actually working. But what happens then to your glucose uptake? Glucose uptake goes up in all of the cells, but even more in the CD11B. So the macrophages who are already taking up a bunch of glucose will take up even more if you take away their glutamine. But the same is true for the T cells and also for the tumor cells. So, metabolism in the tumor microenvironment. So this is our simplified model. If the only cells that were there were cancer cells, macrophages, and T cells, we have to further reduce this as we move forward. But the glucose promotes, but glutamine suppresses effector Th1 and CTL, cytotoxic T cell differentiation and function. Glutamine uptake in T cells restrains glucose metabolism and prevents exhaustion. And so if we have glucose going to the macrophages and glutamine going to the cancer cells, it is no wonder that the T cells are not very happy. That glucose, as we said, is mostly going hematopoietic, while the glutamine is cancer cell specific. And the glucose competition is modest, and the FDG reflects how much inflammatory component there is, at least in these varied models. So this is where we are. This is the Rathmell labs trying to find our way. This is the real village of Rathmell in North Yorkshire, which lies between Giggleswick and Wigglesworth. But that aside, finding the way VHL loss, central to clear cell renal cell carcinoma and HIF expression, can reprogram tumor metabolism, but it only applies to tumors that retain HIF1. FH is important for oxidative metabolism and leads to critical glucose dependency. Separate arm of the lab's trying to understand FH. But in spite of the cancer cell metabolic changes of VHL loss, there is plenty of glucose in the tumor microenvironment for the neighboring cells. FDG PET scans, at least in these models, and we're working on trying to know how much the supply is in human tumors, reflects the metabolism of cancer cells as well as inflammatory components, probably more so inflammatory components. And that macrophages are the major consumers of glucose in across tumor models, and that glucose consumption is increased in all cell lines when you limit or restrict glutamine uptake. And that can also enhance T cell recovery and activation. So there are a million things going on to try to dive into this deeper. We really feel like we've just cracked into this and need to understand it a lot more. But we want to understand how much the tumor cell mutation profile influences the tumor microenvironment. So handily, we have two similar but not the same mutations. So how does VHL mutation versus FH change the tumor microenvironment, the macrophage or the T cell metabolism? Are the T cells in renal cell carcinoma tumors really truly recoverable? We've shown we can do this in vitro, but is this a route to make immunotherapy work better? Or do we need strategies that bring fresh T cells in to the tumor? And can we understand macrophage biology or metabolism as a better way of harnessing immune response? And can we understand our lipids and cholesterols as well to help understand this too? And so with that, I'll end. This is the lab. The people in red, I think I highlighted as we went. I missed some old work. The old FDG PET study was Samira Brooks. The old HIF1 overexpression, HIF2 overexpression in mouse kidney model was Alex Ariel. I mentioned everyone else as we went through, but we're having a lot of fun with this area, and I'm happy to take questions.
Thank you so much, Kim, for that wonderful talk. I now recognize Brad Reinfeld as somebody who's on Twitter, which is great. I do have a question for you as you've studied the metabolism in the immune compartment versus the tumor cells. What are we learning about the impact of modulation of either glucose metabolism or glutamine metabolism on just the immune makeup or trafficking of immune cells?
Yeah, there's not a lot of work that's been done on that, to be completely honest. Most of this work has been in rheumatologic disease and less so in cancer. The first generation glutamine synthase inhibitors like CB839 has been tested in patients and was not markedly positive. But I'm not at all dissuaded by that. I think it may be one, the first generation drug isn't the one that's really bringing the T cells back, and maybe we didn't induce the T cells in the right way at the time that we were pushing them with the glutamine, or with glutamine restriction. So I think that there's a lot of interesting work to do there. But to be honest, I mean, there's a bunch of work with glutamine inhibitors, a little bit with GLUT1 inhibitors, and I don't know that anybody's gone in and said, and what happened to the immune cells? It's always really been very focused on the cancer cells. And I'm guilty of that as well for a long time. You know, way back, I don't know, 2010 or so, Amato Giacchia did a screen for sort of a synthetic lethality with VHL in kidney tumors, and GLUT1 inhibitors came out then as well. So I think GLUT1 inhibitor could potentially be really valuable, both in the cancer and modulating the immune cell at the same time. But I think a lot of work to do in this space.
Thank you. Other questions? Just go ahead and unmute yourself, please. Dr. Quisenberry, you have to unmute yourself.
While he's unmuting, I will say that one other thing that we're really excited to do is to do an FDG PET and a F18 glutamine in patients, and really see what's happening with that. So,
in looking at your macrophage populations, have you subsetted them into inflammatory macrophages and kind of good macrophages?
Right, not well. I mean, we've really done CD11Bs and F480s as much as we've gone down that road. We haven't even really gone to calling them M1 and M2. I'm new to macrophage biology. I really think there's going to be a whole bunch of different kinds of macrophages in there. And so I don't feel like I know which ones are functioning in an inhibitory or which ones are stimulating somebody. So
we're just getting into it and beginning to see that these may play a major role in pulmonary hypertension.
Yeah. Jeff's lab has a new macrophage biologist working in the group, and so we're really excited to work with her. So,
other questions? You can also use the chat.
That's okay. There's more work to do. I'll see the next steps.
Yeah. We thank you so much for the presentation. You know, there's so much to learn and so much to do, and we'd obviously love to have you back as you continue to learn more in this fascinating direction and developing new therapeutics. So thanks so much for joining us, and have a great afternoon, everybody.
Thanks very much. Great to be here. I'm happy to come again in person sometime.